Hello everyone, welcome to Just Biotech Geeks. I hope that you would have watched part 1 of molecular docking, in which we have demonstrated the procedure for protein and ligand preparation. In this tutorial, we are going to see how to set the grid parameters for molecular docking. I would like to mention that I will be providing the link to download all the softwares as well as the websites required for molecular docking in the description of this video. Let us begin by copying the prepared protein structure to a separate folder. Now, let us copy the ligand structure to the same folder. Here we have copied both the prepared ligand and protein to a folder. Now, I would be showing how to install MGL tools for setting the parameters for molecular docking. First of all, you must download MGL tool software in your system. The link for the software will be provided in the description of this video. Here, I have already downloaded the software. Now, let us double click on the setup file. Now, click Yes. Click on Next. Click on I accept and click on Yes. Click on Next. Here the installation process has started. This might take few minutes for installing MGL tool. Once the installation process is complete, you can see a dialog box. Click on OK. Click on Finish. Here, we have successfully installed MGL tools on our computer. Now, we can see the interface of the software. For time being, let us close this software. Next, let us go to the search bar and search for Autodock tools. Now, we must click on the file. Click on Read Molecule. Go to File. Go to the folder where you have saved the protein file. Select the particular protein and click on Open. Here, you can see the 3D structure of prepared protein. By clicking on this arrow, you can see the amino acid present in the particular protein structure. You can also change the view of protein structure in surface format just by clicking on this button and deselect it to view in normal format. Select the next button to view in ribbon format and deselect it to view in normal format. You can also view it in sphere format. As hydrogen atoms are very small, there is a high chance that some hydrogen atoms are missed during protein structure determination during NMR spectroscopy or X-ray diffraction method. So, we must add hydrogen atoms manually using MGL tool software. For adding the hydrogen bond, click on Edit. Go to Hydrogen, click on Add, select Polar only and click on OK. Now, hydrogen bond is added to the protein molecule. This will be favorable to us to find binding affinity of ligand against the protein. Next, we must add charges to the protein structure. So, for adding charges, click on the Edit option. Go to Charges and select Coleman charges and press OK. Here, Coleman charges have been added to the protein structure. Next, we must go to Edit and click on Atoms and we must assign AD4 type to this protein. Now, this protein is ready for further processing, so let us save this file in PDBQT format. For saving this protein file, go to File, click on Save and then select Write PDBQT. Select the location where you want to save this particular file. So, I will be saving this particular file in the molecular docking folder that I have already created. You may decide the name of your choice for the protein, but here I will be naming it as 2fk4.pdbqt. Make sure that you don't leave any space while naming the file. This can lead to an error while running the file. Now, click on Save. Here, we must select the available PDB records from atoms till end and click on Add. Now, all the records have been added. Now, click and select Sort Nodes. Next, click and select Save Transformed Chords. Now click on OK. Here you can view the protein file in PDB-QT format. Next, click on the ligand and select Input and click on Open and go to the folder where you have saved the prepared ligand. Now here, you are not able to view the ligand file. So make sure that you change the file type to All Files. Now you can see the particular ligand file over here. Select this ligand file and click Open. Now, gesture charges and 18 non-polar hydrogen bonds have been added automatically. This might change depending on the ligand. Now, click on OK. Now, use your mouse to rotate the 3D structure, and here you can see the ligand structure have been added successfully. Manually, it is difficult to identify the bond angle of the ligand molecule, which gives the best result in binding with the protein. So, here we are using the software to identify the position. Now, go to ligand and click on torsion tree. Click on detect route. Now, again go to ligand, go to torsion tree and click on choose route. Here, the particular route have been selected. Now, we must go to the ligand and click on output and save it in PDB-QT format. Make sure that you select the particular folder where you have saved the previous file. I will be saving the file in the molecular docking folder. Now, name the file as per your choice. I will be naming the file as 1.pdbqt and click on save. Here, you can view the ligand file in PDBQT format. Next, let us fix the grid parameters in the protein structure for docking. So, before setting the grid parameters, we have to find the active sites where we can fix the grid. To find the active sites, we must go to Google Chrome and search for MetaPocket and go to the website. I will be providing the link to the particular website in the description of this video. MetaPocket is a website that is programmed to identify the active pockets in the protein structure using different algorithms. Now, let us click on Home and just enter the PDB ID of the desired protein in the search box as 2FK4. Now, let us keep the number of pockets as 3 and click on Find Pockets. This might take some time to run and identify the active site. In the meanwhile, let us set the grid parameters. So, for setting the grid parameters, we must go to Grid and click on Macromolecules and click on Choose, and now you must select the protein and then click on Select Molecule. Now you must press OK and navigate to the folder where you want to save the file. I will be saving this file in the molecular docking folder, and now as you can see already a protein file is existing. We must name our current file as the same, that is 2fk4.pdbqt, and rewrite the older file. Now click on Save and press Yes to rewrite the file. You must set the ligand parameters. So for setting the ligand parameters, go and click on Grid. Click on Set Map Type and click on Choose Ligand and click on 1 and then click on Select Ligand. Here your ligand is being selected. Next, we must set the grid box. For setting the grid box, go to Grid, click on Grid Box. Here, you can see a dialog box with default values. The number of points in the X, Y and Z dimension is set as default. So we can change the points of grid just by clicking and dragging over the button. Basically, if you know the grid area of the protein molecule, you can drag the point accordingly. If not, you can keep the default parameters as 40. So here, I would be dragging the X and Y points and setting it to 72, and Z I will keep it unchanged. Next, here we can see the center grid box. For setting the coordinates of X, Y and Z, we must click on center and pick an atom. Now, it is set to default. Next, Let us go to Google Chrome and check the MetaPocket site, which we have initiated to find the Pocket site. You can see the process is still running. For getting the result, I have performed this same process earlier. It took me around 10 to 15 minutes to get a result. As you can see, I have opened the result in a new tab. Now, I will be using the coordinate values to set my grid parameters. We can either copy the value of x, y, and z coordinates directly or download them by clicking over here. I will be just copying these values of x, y, and z coordinates one by one and paste them into this box. Here our coordinates have been set. Now let us click on File and select Close Saving Current. Next, we must go to Grid and click on Output and Save in GPF format. Select the folder where you want to save the file. I would be saving the file in the molecular docking folder. Now, name it as 1.gpf or any name of your choice, but make sure that you don't leave any space while naming. Now, you may click on Save. Here you can see that 1.gpf file have been saved. So far, we have assigned the coordinates for docking. Now, let us fix the algorithm for molecular docking process. Now, let us go to docking, click on macromolecules and click on Set Rigid File Name. Now, go to Molecular Docking folder and select the 2fk4.pdbqt protein and click on Open. Again, go back to Docking, click on Ligand Molecule, click on Choose and select the ligand molecule. Click on Select Ligand. Here, I will keep the parameters unchanged and then click on Accept. Next, let us go to Docking again and set the parameter and set Genetic Algorithm. Here, the default value assigned will be 10. But for more accurate and best result, we must run at least 500 to 1000 runs. I would like to make one point very clear that if we set 500 runs, the program might take around 5 to 6 hours at least to complete the run during the docking process. I will be demonstrating in part 3 of molecular docking. So, while practicing molecular docking, you may set it as 10 or 20 GA runs. As this is a project which I am working on, I will be setting it as 500 GA runs for better result and keep all other parameters unchanged. Now, click on Accept. Next, I am going to save this file. Click on Docking, go to Output, save it as Lamarckian GA. Now, select the folder where you want to save this file. I would be saving this file in the molecular docking folder. Now, name it as protein.dpf or any name of your choice. Now, click on Save. Your file is being saved in the folder in DPF format. Now, we will be using both the GPF and DPF file for further analysis using Sigwin terminals. That's all for this video. Watch part 3 of molecular docking using Sigwin terminals. Do like, share and subscribe to our channel.